how is health affected? Now, next page, turn the page number 146. You can write there itself. What is health? What are the factors affecting health? Again, they have given there, so you can number them. Number one, balanced diet. Number two, personal hygiene. Number three, regular exercise. Did you get it? All of you all are getting the pages 146.
Yes, ma'am.
Okay, so keep noting the things and keep numbering. Children, this year, each and every line of the textbook you have to go through very nicely. From any line, any page, the questions can be framed. And so you keep numbering the balanced diet, number one, then personal hygiene, regular exercise, vaccination. When you come down, fourth line, vaccination, that is your fourth factor. Did you get it? Now, my next question, what is a disease? Write the differences between the infectious and non-infectious diseases. Now we come to what are pathogens. Name the common pathogens. Then the one by one we'll start with the infectious diseases. So it is what is typhoid, name its causal organism, how the causal organism of typhoid enters the body, what are its symptoms, and how can you prevent typhoid. What is the diagnostic test for the typhoid? Now you come to pneumonia. What is the causal organism of pneumonia? How the Streptococcus pneumoniae enters the body. What are the symptoms of pneumonia? How can you prevent pneumonia? Then name other bacterial diseases. Then other one, what is common cold? Name its causal organism. What are its symptoms? How can you prevent common cold? Then the malaria. So we have covered up all questions related to malaria, the life cycle we have done. Now comes your amoebiasis. So amoebiasis, again, very important question, that how can you prevent amoebiasis? Then name the causal organism of amoebiasis, how the ant amoeba enters the human body. Then same way, ascaris. What is ascaris? Which disease is caused by ascaris? To which category, to which group of organism does it belong? Then what is elephantiasis or filariasis? Name the causal organism, its symptoms. Then ringworm. Name the causal organisms of ringworm. So you can see here in your textbook that three causal organisms are given. I want Raj to quickly read out the page number 149. I hope, Raj, you have the textbook in front of you.
Yes, ma'am.
Okay, so quickly name the causal organisms of ringworm. Be quick today, we have to complete the immunity. Fast now.
Microsporum,
Yeah. So keep numbering. Number one, microsporum. Then number two,
trichophyton,
trichophyton. Okay. Then number three,
epidermophyton,
epidermophyton. So Raj, write it in your book itself. Number it. The first organism, the microsporum. Second organism, trichophyton. The third one is epidermophyton. Now they have given very nicely what are the symptoms of this ringworm. So it causes, so already you have noted down all that. And now we come to the next very important topic, very important question, that how can you prevent the infectious diseases? How can you prevent infectious diseases? So they have given here the maintenance of the personal and public hygiene. That is important for checking the communicable or the infectious diseases. Okay, then consumption of the clean drinking water, then proper disposal of the waste. So I want everyone to read this last paragraph and try to make the points, try to learn it in points that what are the methods for preventing the infectious diseases. Then when you turn the page, you find that for the diseases like malaria and filariasis, what have to be done, what precautions should be taken, what preventive measures should be taken so that these two diseases are not transmitted. So they have given by avoiding the stagnation of the water. Are you all getting, children? Everyone following in the textbook or not?
Yes, ma'am.
Yes, ma'am.
So try to make the points there. Okay, and you will prepare from there only. Number one, what? That is by avoiding the stagnation of water in and around the residential areas, regular cleaning of the household coolers. So keep numbering. That is very, very important, children, for remembering later for the examination. And when you write the answer, I want that in the points only. Use of the mosquito nets, introducing the fishes like gambusia in ponds. This is very important. It is asked for short question that how can you prevent the malarial parasites. Okay, so it is introducing the fishes like the gambusia in ponds that feed on the mosquitoes' larvae, then spraying of the insecticides in ditches. So these are the methods which are checking the vectors, children. And once you are checking the vectors, so then multiplication of vectors will be checked. So then the transmission of that particular pathogen will also be prevented. Then drainage areas, did you get it? The spraying of the insecticides in the ditches, drainage areas, and the swamps. And doors and windows should be provided with the wire mesh. So you know, in our houses we are having these wire meshes. Now the other one they have given the vectors, that is the Aedes mosquito diseases like the dengue and chikungunya. In they are the vector borne. So this examples more you have to write. You have to remember. Apart from that, the vaccination. One very important preventive measure is if you want to check the diseases, so we are doing that only, children. To prevent ourselves from the COVID, now the vaccination has started. So other method of prevention is vaccination. So they have given the use of vaccines and immunization programs. They will enable us to completely eradicate the deadly diseases. So already we have eradicated number one, polio. Which other disease has been eradicated? Say it quickly.
Polio and smallpox.
Okay. Everyone knows. So now this was the topic which we have already completed. All these questions everyone will prepare. Now we will start today with the immunity. So now you have seen that the next topic 8.2 is immunity. So what you do in your notebook, write down immunity.
Ma'am,
One, yes, yes, Neha.
ma'am, you said that we have to. You will give us the practicals.
Acha, acha. Okay, so time first we will give for the practicals. Okay, so everyone can start. Now, do you have the lab manual in front of you?
Yes, ma'am.
Okay, so section A, read it out. How many students are having the lab manual? Pick up. Shravani, you have?
No, ma'am.
Okay, तो जिसके पास नहीं है वो बता दो कि I don't have. So then,
I don't have.
I don't
Neha,
have. I also don't have.
okay, so that means how many are having? Devanshi, you have?
Yes, ma'am.
Okay, Devanshi, Bharvi, and Neha. Only three students are having. Okay, so what we'll do, then I am telling Neha will mark it and she will WhatsApp you. And as you buy the lab manual, you can start your one. It will be okay?
Yes, ma'am.
Yes, ma'am.
Okay. So Neha, now you can read out the section A, first aim.
Ma'am, first is to study the pollen germination on a slide.
So everyone will record this experiment. You can mark it, Neha. Okay, to study the pollen germination on the slide. So as it is given in the lab manual, everything you have to copy. Okay, and figures which are drawn in the textbook, you have to draw all the figures on the drawing side. Okay, now second experiment.
Uh,
So this is
Study of
section.
physics.
Okay, section A, you will, as you did last time, you made one page where you wrote section A, then you started with the section A experiment, same way you do it. Okay, so this will be experiment number one, pollen germination on slide. Second one,
Study of physical properties and pH of different soils.
okay. So are you reading from the name or the content of the book, Neha?
Ma'am, I'm actually reading from the contents only.
Contents, okay. Syllabus part you read it. Where the syllabus is given now?
Ma'am, like I have to read like the both the experiments that is included in that aim.
Right. Eight.
Okay, ma'am. So the first is to study the texture and moisture content of different soils, and
Uh,
second to study pH of different types of soil.
okay. Now see children, what you'll be doing, you will write down the common aim. And common aim which is given is to study the pH, the texture, water holding capacity and moisture content. Okay, four things you have to study in this experiment, but where in the lab manual you will find it must be given separately. So, but you will put the common experiment number. You can write down the aim to study the pH, water holding capacity, the texture also, pH, texture, then water holding capacity and moisture content. Moisture content of the different soil samples. तो you just remember बाकी aim आप book से copy कर सकते हैं. Remember four things you have to do. Okay, four. Number one, that is your texture, then the pH order can change. Okay, pH, texture, water holding capacity, and the moisture content. Now, when you see it in the textbook, I think you that is your lab manual, you will find that two together are given, na? Texture and water holding capacity, Neha.
Ma'am, texture and moisture content is together, then pH and water holding is another aim only.
Okay, so you make it as A and B. What you call of your
Yes, ma'am.
A and B it will be in your file. But one common in experiment number two, one common aim you put. Then write down experiment number A, so you can take it as the first one as it is given in the lab manual. Okay, now third experiment.
Ma'am, it has three parts. Third one, first is collect water from two different water bodies around you and study them for pH. Second, collect water from two different water bodies around you and study them for clarity and presence of particulate matter in different samples of water. Third, to study different water samples for the presence of living organisms.
Okay, now you make it one experiment, children. Okay, this will be experiment number three, that to study the clarity, presence of particulate matter, the pH, and the presence of living organisms in the different water samples. Did you get it, everyone? How you will combine all this water samples one all together, you'll write down experiment number three. Later you can split up. Is it clear, Neha?
Yes, ma'am.
So read out again what will be the aim.
To study the pH, clarity, presence of particulate matter, and presence of living organisms in different water samples.
Water samples. Everyone has noted? Yes or no? अनम्यूट करके बोल सकते हो आप लोग.
Yes, ma'am.
Rumisha. Okay, what about Rumisha, Raj, Ritu?
Yes, ma'am.
Yes, ma'am.
Ah, now the next experiment.
Ma'am, to study population density and percentage frequency of different plant species of a given area.
Ah, so here again, remember two things are there. Okay, you have to study the population density and frequency, children. So one common aim will be there, and later then you can write down A and B. Because population density, you will find the table will be there, then separate they have given. Okay, so according to the lab manual, what they have given, if they have split up, you write it A and B. Otherwise, you can do it separately, means in one also. Okay, whatever is given in the lab manual. Neha, just check it in lab manual. They have separated the two population
Ma'am, this one is combined.
combined. So then no problem. You will do it together only. Okay, then next one.
To prepare temporary acetocarmine stain mount of onion root tip to study various stages of mitosis.
Mitosis. Okay, so it will remain as it is. You will write also everything, whatever is given there. Now, for the population density one, children, the diagrams which is given there of the quadrant. Okay, few extra diagrams are given. That one, so I'll tell you, you record, children, three experiments. One is your pollen germination. There no changes will be there. Whatever is given in the lab manual, you'll do it as it is. Second experiment is your soil related, na Neha?
Yes, ma'am.
Huh. So soil related also you will do everything, but one change will be there, children, in the water holding capacity. You just open that experiment, Neha. Devansh, you also have the book and Barbie. You
Yes,
can
ma'am.
just look at that experiment. उसमें देखो आपका एक for the water holding capacity ना they have given the measuring cylinders. Did you get it?
Yes, ma'am.
Measuring cylinders with the funnel.
Yes, ma'am.
Okay, so that one we will do. That procedure we will be doing, adopting. Okay, in that case, in water holding capacity now you will observe two are there. Do you have three in your lab manual? Are you having comprehensive now?
Yes, ma'am. Comprehensive.
Okay. So you will find in one petri dish is there and then small beaker kept in that. Is it there?
Yes, ma'am.
So don't do that one. For
Okay,
now,
ma'am.
which procedure we will be adopting?
The measuring cylinder one.
Measuring cylinder with the funnel and all. Okay, so there they must be shown two. So
Hmm.
that one will do it in the file.
Yes, ma'am.
Okay. Now for the moisture content, you must be seeing there the a tripod stand must be there with the china dish on top. Is it there that again
Ma'am, which one?
for the moisture content? Did you get it?
Ma'am, just a minute.
Devanshi got it?
Yes, ma'am.
Yes, ma'am.
The tripod stand on that you find the china dishes kept and that the soil is being heated. So that is for the
Yes, ma'am.
moisture content. So that one you will be doing. Okay, and pH to no diagram will be there. Okay, and for texture you must be seeing there the beaker is there with the layers of the soil. Is it there?
Yes.
Yes,
Yes,
ma'am.
ma'am.
So that diagram you will draw. Understood, everyone? So you complete the first and that is the pollen germination, and the next one will be your soil related.
Ma'am, the soil one.
Soil related. Okay. So these two you complete very nicely. You write everything whatever is given, and procedure बस उसमें ये देखना है कि दो procedure दिए आप एक करेंगे. Is it clear? And rest you will copy the precautions and everything and figure also related to that only. Okay.
Yes, ma'am.
So these experiments, two experiments you record it and show it to me. On which day? Not tomorrow, day after tomorrow, we will not have class. Monday. It will be done. 2 days okay for you all?
Yes, ma'am.
Yes, ma'am.
Okay. So now we will start with the
Ma'am, ma'am, in the
yes.
soil one, we have to write pH one in the
Soil
second
pH you
experiment.
have. Yeah, pH, so you have to do. Because four things we are doing in the soil. We'll check the pH. Okay, we will be doing the texture.
Moist.
So for texture only, they have shown that beaker, and then you have to dissolve it, and all layers will settle down. So we'll come to know that how much is the sand, how much the silt is there. Okay, so that they have
Yes,
shown
ma'am.
in the manual. That one?
Ma'am,
Yes, yes. Yes, Rumisha?
ma'am, can I show you on Wednesday instead of Monday?
Okay, Wednesday. Okay. You'll take time in purchasing the book.
Ma'am, because from today evening there is a lockdown, complete lockdown, so it won't be
Right.
possible.
It will not be right. Okay. So if you buy today only तो हो सकता है नहीं तो फिर तो 2 दिन कुछ शॉपिंग नहीं हो पाएगी आपकी. Okay. So you can show it to me on Wednesday, no problem.
Okay, ma'am.
So those
Ma'am,
students who can arrange, those students who can arrange, it's okay. Otherwise, show it to me on Wednesday.
ma'am, so I have to send the pics of only the contents or the inside everything.
अच्छा हाँ, that can also be done. Rumisha, what you say? If she sends you, तो then papers का problem होगा, file papers का. Isn't it, Rumisha?
Yes, ma'am.
हाँ, तो अगर फाइल पेपर्स अरेंज हो सकते हैं तब तो फिर अगर नेहा इज सेंडिंग द पीडीएफ ऑफ दैट ओनली द पेजेस ऑफ द लैब मैनुअल तो फिर लैब मैनुअल ना भी हो तो भी चल जाएगा काम. इजंट इट?
मैम
तो
पेजेस का मैं ट्राई करती हूँ अगर आसपास कहीं मिल जाता है अभी.
ओके, तो नेहा यू कैन सेंड इट, नो प्रॉब्लम इन दैट.
Okay, ma'am.
हाँ, so their problem of that lab manual will be solved. Okay, just buy the pages. If it's not available, if the lab manual is not available, 5 pages तो easily available हो ही जाते हैं. Okay, so you can complete it. And then one more thing, children, what you do, see the investigatory projects given. Okay, and any investigatory project, whichever you like, select one. Okay, so next period I'll give you the synopsis as you did last time, and that also you can start. Okay, so April month में आपका investigatory project भी हो जाए और section 8 पूरा complete हो जाए April month. Is it okay to you?
Yes,
Yes,
ma'am.
ma'am.
ओके, सो इन्वेस्टिगेटिव प्रोजेक्ट का पीडीएफ जैसा अपन ने लास्ट टाइम किया था वैसे आप कर लीजिए. सिनॉप्सिस आई गिव यू, यू सेलेक्ट वन टॉपिक. ओके, so फिर after that you will just concentrate on the theory part. Okay, so shall we start with that? I'm presenting my screen now. So we will start today with the immunity. Now children, everyone knows that today some people are suffering from the corona, the COVID-19, and some people are not suffering from that. Some are, if it is infected also, if someone gets infected also, you find some are getting cured very easily and coming back home, and some are not able to. What is the reason behind it? What is the reason behind it, children?
Ma'am, some are
Immunity.
having immunity, strong immunity.
Some are having the resistance power, so they are able to fight. Okay, and some people are not having the good immunity, the resistance power is less, so that's why they will have to, they are not able to recover with it and leading to the death. Now, so immunity is very important part of our body, children, which is helping in the survival. Because all around in the environment, you know that we are exposed to so many microorganisms. So we are moving. We cannot save ourselves from the different pathogens entering the body. Only we can prevent, we can protect our body by making the immune system strong, so that if any harmful organism enters the body, to then our body, the which cells are fighting with that? Do you all know?
WBC.
WBC.
WBCs. Okay, but WBCs are producing the antibodies. What the WBCs produce?
Antibodies.
Antibodies. And these antibodies fight with those pathogens and destroy them. So we will do it in detail, children, how this immune system works. Okay, the immunity, the resistance power is developed by one system of our body, and we call that system as the immune system. And there are many components of the immune system. One by one, we will try to understand. You can note down what is immunity. It is the disease resistance. It is the disease resistance, that is the ability of an organism to resist the diseases or disorders. Noting? Noted, everyone?
Yes, ma'am.
Okay. The Now, what is this branch of biology called which deals with this study of immunity? So it is known as immunology. Raj, what is that branch of biology called which studies about this all immunity?
Immunology.
Immunology. Okay. And the person who is not susceptible to a disease is immune to it. What we say, that person is immune to such a such disease. Now, when you are taking the vaccine of the COVID-19, means you are getting immune to the COVID-19. Okay, now immune system is the body's line of defense against pathogens. So all vertebrates, that is the higher vertebrates, they have a complex immune system, and that immune system produces the antibodies, and these antibodies are specific to the foreign bodies. Okay, and that is the uh, main reason why we are able to make the vaccines. Okay, because vaccines are helping the body in fighting with that particular foreign body. Okay, if the polio vaccine is given, you find that polio vaccine, once it is given, the body's immune system starts making that particular antibodies which will fight with that polio virus, and it will remain in the body throughout the life as memory cells. Remains in the body throughout the life as memory cells. So, what is the important feature of this immune system? That can even remember attacks. The immune system can remember attacks by a particular pathogen and prepare for future infections. So, how it is preparing for future infections? By making the memory cells. Noted, everyone?
Ma'am, wait.
Huh. Immune system can remember the attacks by particular pathogen. Written. Now we start with the types of immunity. So quickly write down types of immunity. So there are two types of immunity, children: the innate immunity and the acquired immunity. The innate immunity is that which we are born with, children, and acquired immunity which we develop during the lifetime. We are non we are not born with the acquired immunity. We develop the acquired immunity by giving the vaccines, as well as when once the child is born, children, so child is exposed to the outer environment, and one by one the different pathogens keep entering the body, and the immune system of the body continues to produce different antibodies, fights with those different pathogens, and then makes the memory cells. That's why you find that we always say that the child should be exposed to the outer environment. Child should be allowed to play outside. And you must be observing there are some people who live in isolation, वो बचपन से जिन बच्चों को एसीज में एक बंद कमरे में प्रोटेक्टेड बहुत रखा जाता है, तो यू मस्ट बी ऑब्जर्विंग वो बाहर का कोई पानी भी नहीं पी सकते, बाहर का कुछ खा नहीं सकते। ओके, उनको बाहर अगर वो कोई ऐसे कंटामिनेटेड एरिया या पोल्यूटेड एरिया में चले जाएं तो दे विल हैव लॉट्स ऑफ प्रॉब्लम। सो व्हाई डज इट हैपन? क्योंकि बचपन से उनको एक्सपोजर नहीं मिला। So that is why those pathogens have not entered in their body और उनकी बॉडी में इम्यूनिटी डेवलप नहीं हो पाई। So what is important for this? That's why in the schools you must be knowing why we want the children to play outside. Why we want you all to play in the ground? Because you know the soil is having many pathogens, and those pathogens will gradually enter your body. And one by one, the body's immune system will make the antibodies against those different pathogens. Is it clear? तो इसलिए आपने देखा बच्चे जब छोटे रहते हैं तो दे फॉल इल अलॉट। जैसे ही वो 4-5 years के हो जाते हैं तो यू फाइंड दैट दे विल नॉट फॉल इल। द रीजन इज व्हाट? तब तक क्या हो जाता है 5 years तक? कैन एनीवन टेल what happens in those 4-5 years?
Immune system develops.
Immune system develops, and immune system develops when you say, remember, the antibodies develop. What you have to say?
The antibodies develop.
इम्यून सिस्टम तो डेवलप होके ही वो बॉर्न होता है। The child is born with the developed immune system, but the antibodies formation will take place only when the exposure is there to particular pathogen. अगर हमारी बॉडी किसी पैथोजेंस के लिए एक्सपोज नहीं होगी तो इम्यून सिस्टम भी एक्टिव होकर उसके लिए एंटीबॉडीज, उसके अगेंस्ट एंटीबॉडीज नहीं बनाएगा। So what is important, children, to make your body strong? What is important? Rumisha, what is important to make your immune system strong and make your body strong?
The exposure of body to the pathogens.
हाँ, एक्सपोजर ऑफ द बॉडी टू द डिफरेंट पैथोजेंस। इसलिए घर में बैठ के एक एसी कमरे में बंद रह के आप सोचे कि हम प्रोटेक्टेड और हम स्ट्रॉन्ग हो जाएंगे, तो दैट इज नॉट पॉसिबल। दैट्स व्हाई children should be allowed to play outside. Is it clear? Okay, now we quickly try to differentiate between the two. Innate immunity. I want Ritu to read out the first difference.
Innate immunity is acquired by the fetus from the mother through the placenta. Acquired immunity is present at birth but is acquired during one's lifespan.
All have written this difference. Did you note down, everyone? Yes or no?
Yes, ma'am.
Everyone make 2 columns and note down. Okay, now second difference with 2. Ritu, second difference.
Innate immunity includes non-specific body defenses against infection. Acquired immunity. It is developed by an organism in response to infections by pathogens, foreign bodies, or when vaccines are administered.
Okay, so I hope it's very clear that the innate immunity is non-specific. Okay, it is non-specific. It is not for a particular pathogen. Is it clear? But the acquired immunity we develop for, or it is developed for the particular pathogens, particular foreign bodies. Okay, and that is done by giving the vaccines. Okay, now Shravani, read out the third difference.
Ma'am,
Shravani,
actually, I'm having some network problems, so the screen is not visible to me.
Acha, screen is not visible to you. Okay, so I want Devanshi to read now. So then, are you able to write down, or then it has to be dictated to write, na?
Yes, ma'am. Ma'am, afterwards
Shravani?
I will see the recording and I will write down the things.
अच्छा, that can be done. Okay, okay, ठीक है, ठीक है. हाँ, but समझ में आ रहा है श्रावणी?
Yes, ma'am. The
You are
voice is
following
clear,
the
but the screen is
हाँ
not
हाँ.
visible.
Okay, so try to understand, okay, that innate immunity we are born with, acquired immunity we develop it during the lifetime. The innate immunity is non-specific, but the acquired immunity is specific. It is for a particular disease, specific for the particular pathogens, foreign bodies, or the different causal organisms, and we develop it by the vaccination process. Okay, now the third one, innate immune responses are similar for all pathogen foreign bodies. The responses will be same. For example, the tear stimulation, for the dust, any other irritant pathogens, this is your, the responses are similar. And when a pathogen enters the body, the acquired immune system generates a primary immune response. It develops the primary immune response. The body remembers the pathogens and encounters. So this I have already discussed. Now the entry of pathogens is checked by certain physical barriers. Now innate immunity, it is developed by the different barriers. So we will try to take up now the different barriers, children. All have noted this? In the acquired immunity, barriers will not work. Okay, acquired immunity is developed by the formation of the memory cells. By the formation of the memory cells. And these memory cells will multiply, will start multiplying very fast at the second encounter of the same pathogen. On the second encounter of the same pathogen. तो अगर एक बार वैक्सीन पोलियो का लग जाता है, अगर at any age of the life if the polio virus enters the body children, तो memory cells are already present in the body क्योंकि वो तो बचपन में दे दिया गया था और इम्यून सिस्टम बॉडी के इम्यून सिस्टम ने मेमोरी सेल्स बना के रख लिए हैं. अगर सेकेंड एक्सपोजर थर्ड एक्सपोजर कभी भी बॉडी को मिलता है, so you will find those memory cells will quickly start multiplying, will make large number of the antibodies, and they will destroy that virus, and the disease will not be caused. Okay, so now we'll take up quickly the barriers. So write down. So innate immunity consists of four types of barriers. Innate immunity consists of four types of barriers. Number one, physical barriers. Physical barriers. Then physiological barriers. It's given very nicely in the textbook also, children. Try to understand the things. Physical barriers, then physiological barriers, cellular barriers, and cytokine barriers. Cytokine barriers. Now try to understand, children. Physical means that physically the pathogens are prevented from entering into the body. So you know that the first one, the skin is there. The physical barriers, we find that the skin is your first line of defense. Skin is the first line of defense. Now, how the skin is preventing the entry of the pathogen? Physically, it is preventing. The next, remember that the skin is having the sweat glands, and the sweat produced is having the enzyme lysozyme. This lysozyme destroys the bacterial cell wall. It destroys the bacterial cell wall. Then you find the mucous membrane. So mucous membrane Where the mucous membrane is present, in your nostrils it is there. Okay, lining of the respiratory tract is there, then the gastrointestinal, you can mark it in the book, children. Urinogenital tract. Urinogenital tract, they all are having the mucous membrane lining. That is, the epithelium is coated with the mucus. So when the mucus is coated, children, what happens? The pathogens, if they are entering, they get trapped. They get trapped there itself and will be prevented from entering inside. Entering inside. All have understood? The tears, the saliva, the gastric juices, they are considered as the next one. Okay, all have noted the physical barriers. You can mark it in the book, the skin. And number two, mucus coating of the epithelium. All have understood? Okay, now Shravani quickly say, what are physical barriers? What are physical barriers?
Ma'am, skin, mucous membrane.
Okay. Now take up quickly the physiological barriers. So physiological barriers includes your, you can keep numbering, that is the acid in the stomach, saliva in the mouth, and tears of the eyes. Acid in the stomach, saliva in the mouth, and tears. So you remember, in the saliva as well as in your tears, the same enzyme is present, that is lysozyme. Lysozyme, lysozyme. This lysozyme enzyme destroys the bacterial cell wall. Destroys the bacterial cell wall. Understood, everyone? So physiological you have understood. The next one is your cellular barriers. So cellular barriers, remember, they are the WBCs. The special type of the WBCs, which is given in your book, you can mark it there. Polymorphonuclear leukocytes, PMNL neutrophils. And the other one, the monocytes. Then the third one, the natural killers. The natural killer type of lymphocytes. So they are all, so keep numbering. Number one, what are those special cells which are acting as the barriers? Which are acting as the barriers. What are they? Number one, PMNL. Then number two, monocytes. Number three, natural killer cells. Natural killer cells. Then number four, macrophages. Number four, macrophages. So macrophages, remember, it is present in the areolar tissue. Okay, and the other cells which we have named, they are all present in the blood. And they all are phagocytic in nature. They are phagocytic in nature. So phagocytic means they engulf the foreign bodies and destroy them, digest them. So they are phagocytic in nature. So this way they are acting as the barrier. And the last one quickly take down, that is the cytokine barriers. You also call it as the chemical barriers. They are only known as the chemical barriers. So the question is asked usually, children, related to the cytokine barriers. They are the interferons. So what are interferons? They are the, write it, they are the proteins secreted by the virus infected cells. They are the proteins secreted by the virus infected cells. and make the neighboring cells resistant to the attack of the viruses. Make the neighboring cells resistant to the viral attack. अब एक सेल को बॉडी में अगर वायरस ने अटैक कर लिया है तो वही सेल क्या प्रोड्यूस करेगा? What it will produce then quickly? Children, say. What is that chemical?
Interferons.
इंटरफेरॉन्स. ओके, तो वही वायरस ने जिस सेल को अटैक किया वही सेल प्रोड्यूस करेगा इंटरफेरॉन्स. एंड नाउ व्हाट दिस इंटरफेरॉन विल डू? इट विल now make the other neighboring cells resistant to the attack of the virus. So this way it is prevented. So it is a chemical barrier. It is a chemical barrier. So we have completed today the innate immunity. Acquired immunity we'll take up next time. I'm stopping the sharing. Okay, so now we'll do Shanti part, children. Try to read everything. Acquired immunity also, try to read it in the textbook. We'll discuss in the next class. So we'll do Shanti part. Om Shanti Shanti Shanti. Thailand, children. So in these 2 days, children, what you will be doing? You will be doing recording 2 experiments. You'll try to read the investigatory projects, whatever you like, you select it for yourself, and try to read the pages of your textbook till the acquired immunity topic. Okay, so Sairam, you can read now.
Thank you, ma'am.
Thank you, ma'am.
Thank you,
Saira,
ma'am.
ma'am.
Sairam,
Thank you,
Sairam,
ma'am.
Sairam.